So nucleic acids, these are our fourth group of macromolecules, the families of macromolecules, and the elements that they contain. Now we've got three parts. We've got the sugar, and that is going to make up, that's going to be why you need carbon, hydrogen, and oxygen. And then the nitrogen base is the nitrogen, and the phosphorus is what is needed for the phosphate. And so that's why all five of these elements are found in every nucleotide. And the nucleotide is the monomer name, and the dinucleotide is the dimer. We don't really, polynucleotide, nobody really uses that term. But DNA is an example, and that is the code of life. RNA is involved in some way in protein synthesis, and ATP is our energy currency. So we'll get into more details on that in Unit 3 when we talk about microbial genetics and microbial metabolism. But we're going to give you a brief discussion of what the structures of nucleic acids are. Oh, and the bonds are called phosphodiester. I know you really wanted to know that, right? But that is technically the name of the... I just knew you wanted to know the name, but the name of the bond is a phosphodiester. So there's a phosphorus, and it goes between the phosphate and the sugar in the backbone of the molecules. So DNA, deoxyribonucleic acid, is the special coded genetic program with detailed and specific instructions for each organism's heredity. So if it is a living cell, it has double-stranded DNA as its code, its genetic code. Ribonucleic acid, RNA, is helper. It helps the translating or protein synthesis and carrying out the instructions of DNA to make the proteins and other aspects of protein synthesis. So nucleic acid structure, you can see the nitrogen base here in the middle, and the pentose sugar and the phosphate. And so those are the three parts you have on all of these. You may or may not have double strands depending on if it's a virus. You may have DNA that's single-stranded, or RNA may have double strands if it's a virus. But generally in cells, we have double strands of DNA and single-stranded RNA. But notice the one difference, okay, so they have some things that are similar and they have some things that are different. So I want to kind of identify some of those differences. So DNA has deoxyribose for the sugar, and RNA has ribose. So that is a definite difference. So the sugar is different. And then DNA has adenine and thymine, cytosine and guanine, where RNA has adenine and uracil and cytosine and guanine. So they have this difference in the bases. But they have, all of them have, DNA and RNA both have a adenine, both have cytosine, both have guanine. And they both have one phosphate per, so like this is one nucleotide, basically. And so they have one phosphate per nucleotide. So that's another similarity. So purines and pyrimidines. Purines are nitrogen bases composed of two rings. So the two ring structures like these are purines. The one ring structure is the thymine, cytosine, and uracil. They kind of look like snowflakes. And the thymine is replaced by uracil, so that makes sense that they're both pyrimidines. But that is something that I probably will have you do in Unit 3, or, well, when we talk about genetics and metabolism, we'll get into more detail on this. But you can see the pentose sugars, they have 1, 2, 3, for 5 carbons. So pentose means 5. And they all have this oxygen that's really bringing the ring together. And then hydrogen, hydroxyl, hydrogen, 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 hydroxyl. What's missing here is that there should be a hydroxyl here. So instead of having hydroxyl, it just has a hydrogen. And so this would be a difference between DNA and RNA sugars in the molecules. So DNA contains all of the nitrogen bases except uracil, and RNA contains all the nitrogen bases except thymine. Nitrogen bases are covalently linked to deoxyribose, which is the sugar. Nitrogen bases are covalently linked to ribose in RNA. So you can see if you look at the strand, it's like sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. And the nitrogen bases are held in the center, and they're held together by hydrogen bonds. So that's a little bit about differences between DNA and RNA. So RNA is used in protein synthesis. Each of the major types of RNA have some job, like the mRNA is what is translated. That's the blueprints for the gene, the copy of the gene. tRNA transports the correct amino acid to the ribosome for protein assembly, and the rRNA is the major component of the ribosome. So they each have some job in protein synthesis and regulation. And ATP is the energy currency. Like I said, it's the energy molecule. It's what we can spend in the cell. And it has adenosine, it has the adenine nitrogen base, which is a purine, has 2 rings. It has the ribose sugar, and it has 1, 2, 3 phosphates. And I want you to notice that these 3 phosphates are very negatively charged, and so they don't want to be together. So this one's fine, but making this bond and this bond, you have to have a lot of energy to put the 2 negatively charged particles together. And these 2 ions are really not going to want to come together without a lot of energy. So when you break the bond, it releases energy, and it is just like the right amount of energy to do the functions within the cell. So it's our energy currency. It's the Goldilocks of the energy molecules. It has just the right amount of energy to do the work in the cell. Not too much, not too little, just right. And that's a picture of the ball and stick model of adenosine triphosphate. So our concept check for this section is to list and describe the 4 main biochemicals. What are the phospholipids, and why are they important in biological, or why are phospholipids important biological molecules? List and describe the 4 levels of protein structure. Why is ATP an important energy molecule for cells? So we'll stop here and then finish up with the last section. This is going to be the end of test number 1. The next section here in chapter 2 really is an introduction to cells, and so we'll be including that information in test number 2, so not in test number 1.